time, why do the transmembrane segments typically assume secondary structure? Okay? So here they are, these barrels or sort of cylinders represent an alpha helix. The arrows represent a beta sheet. So why were the transmembrane segments mostly alpha helices, sometimes beta sheets? How come I can't just have like a straight line of hydrophobic residues go through? Yes, Matteo. That is a perfect answer. The peptide backbone requires hydrogen bonding. The peptide backbone is polar. Okay? Here's our peptide bond between two adjacent amino acids. It has now polarity to it. It has a hydrogen bond acceptor site and a donor site. In order to satisfy hydrogen bonding that the backbone needs, the backbone's going to interact with itself through a helix or a beta sheet to get across. And here are two additional nice resources that I found that I like that talk about this. Again, I put as many resources as I can there. I encourage you to take a look at them. If you don't like them, that's fine. I mean, if it doesn't work for you, but my explanation of everything isn't always the best. This one is sort of kind of like a walkthrough tutorial. This one's more of like a true 3D video where they're spiraling it. And so I just want to point you to things that I think are nice. Use them if you see fit. Okay. So here, the distance across the membrane we've been saying is roughly about 35 angstroms. When we look at our helix here, our alpha helix, so we have all our side groups on the outside, and there are 3.6 residues per turn. Okay. So each amino acid rotates about 100 degrees. I don't need you to memorize exactly these numbers. I think last time we said each amino residue contributed about 1.8 or 1.75 angstroms for this helix. 1.75 makes the math really nice, so we'll use that one. Remember that these structures are flexible, right? They have the ability to kind of flex and move and stretch in space. So none of these are like hard distances here. But if we think about 35 divided by roughly 1.75 angstroms, sort of per amino acid as you go up the helix, you would require greater than or equal to 20 residues to get across the bilayer. Okay? And so, again, I don't care so much if you remember, 1.5 versus 1.8 versus 1.75, but more like roughly 20 to get across is good. Okay, so 3.6 residues per turn. If you plot each residue every 100 degrees, you can assemble a spiral. Okay, so this was our helical wheel. The value of a helical wheel for us is in the title. It's predicting a helix location in a protein. The reason we care about building a helical wheel would be so that we could figure out where we think this protein is, where we think this helix is within a protein, and then possibly what function it has based on where it's located. So in this particular one here, we start at 1, we move 100 degrees, plot number 2, 3, 4, all the way around. And if we can draw a straight line through right through the center and have all our hydrophobic residues on one side and all the polar or charged residues on the other side, we would describe this helix as amphipathic. So one side of the helix is polar, one side of the helix is nonpolar. Okay, and again, I said this only because there were people who just couldn't keep it straight in past semesters. The helix here, the inside is not an inside. We're going up and down. And this is another link that I also like that covers some of the relevant stuff here in amphipathic helices. So again, the value of a helical wheel is to help us predict location. When we have an amphipathic helix, which is the most common type, and we think of a globular protein, so this is a soluble, completely folded up on itself protein. It's not embedded anywhere. It could be free floating inside the cell or wherever it may be. Okay? We have amphipathic, we'd expect it to be on the surface so that its hydrophobic residues can face the inside and its polar and charged stuff can interact with the water around it. If we had something fully hydrophobic, we'd expect buried in the core of a globular protein. And if we had totally or mostly charged residues, we'd expect it to be fully exposed. So this was for globular proteins. These two are also options for transmembrane. So buried kind of makes sense. If you had totally hydrophobic residues, you expect them to be interacting with the lipid bilayer to get across that distance. But potentially, you could have an amphipathic helix where rather than having the polar side to the outside, you have the polar side to the inside. Okay? Like a channel or a pore. These transmembrane proteins are usually functioning to get stuff across the membrane. Okay? So we could have a nonpolar side sort of facing the hydrophobic residues, and the polar side could be sort of towards the sort of channel going through. Yes? So this is where it depends on if you, and you might not know this, if you have just some brand new structural protein and you're trying to figure out what it does and where it is, you might not know. Maybe depending on where you isolated it from, if you took a cell and lysed it up, or if you just pulled it out of blood or something, you might get a better idea. But if you have a globular protein, totally hydrophobic, it's got to be on the inside protected. If you know that it's embedded in the membrane, then totally hydrophobic could be embedded in the membrane. There's a chance that amphipathic could be here too. And I bring this up because I think it's on the homework number 2. So I'm not sneaky, I'm very helpful. Okay? But I want you to fundamentally understand why this is correct. Okay? So key principle for proteins. Sequence will dictate what structure it's in, will dictate what function it has, and that's going to be tied to what location it's in. Okay? And so if it's embedded in the membrane, maybe it moves stuff across the membrane, maybe it doesn't. Okay? It could have a lot of possible functions. And globular proteins also have a ton of possible functions. They might bind to another protein and form a complex to facilitate transcription or translation. They might bind to DNA and stop it from doing something. Who knows? There's a ton of possibilities. But all I want you to be able to do is look at the sequence and make educated guesses about what that protein could be involved in. Okay? So myoglobin here is an example of our globular protein. So it's water soluble. It has shorter helices. We don't need as long of helices. And we don't really have a lot of nonpolar stuff happening in a row. Whereas bacteriorhodopsin is a transmembrane protein. It does have longer helices, so that's also kind of a clue for us, maybe transmembrane. It needs to get across the thickness of the bilayer, and it's mostly nonpolar. Okay? Or this particular helix here, A, is mostly nonpolar. So in addition to the helical wheel, another thing that we can look at is something called hydrophobicity or hydropathy index or scale. You might see all of these words depending on what resource you're looking at. And this is, again, a measure of how likely something is to be transmembrane. Okay? The hydrophobicity index or scale is calculated by looking at the energy change from moving an individual amino acid from water into something nonpolar. Okay? So if you have something nonpolar, such as tryptophan, and I'll populate the scale on the next slide and it'll be easier to talk about. But if you're moving something that's very hydrophobic, say tryptophan, it doesn't want to be in water. It does want to be in an apolar solvent. When you move it to the apolar solvent, you're going to have a negative change in energy. So you'll have a very high negative score. Okay, so this means hydrophobic stuff over here, whereas stuff that's very hydrophilic, if you have your charged groups, your lysine or your arginines, this stuff does not want to come out of water and go in an apolar solvent. And when you do that, it would be a positive change in energy. It'd take energy to make that happen. Okay, and so it'll fall on this sort of side of this figure. Okay, so one of the questions from last time, true or false, the alpha helix propensity scale and the hydrophobicity scale reveal the same information about amino acid residues. What's the answer? False. Great. Okay. Amino acids have different propensities or desire, willingness, ability to be, preference to be in an alpha helix based on things like their size, their shape, their charge, and their sterics. Okay? Small stuff like alanine's the second smallest thing we've got. It's really easy. It's not charged. It's happy to be in an alpha helix. Glycine's a little bit too flexible to be in an alpha helix. Proline actually is bonded into the backbone, so really doesn't want to be in an alpha helix. Okay, so this is sort of a relative scale, and it depends on all of these things, whether it's charged, what size and shape it has. The hydrophobicity scale is the energetic cost to move the amino acid from water to hydrophobic environment. Okay, it's just a totally different metric, and it's almost exclusively tied to charge. It does not really have anything to do with size or sterics. Okay? And that's repeated here. Hydrophobicity scale describes energetic cost of moving this amino acid from water to a hydrophobic environment. It depends on the side group's polarity or charge, and sterics are not a factor, whereas sterics are clearly a factor for alpha helices. Okay. So this is a slide we didn't get to last time. You don't need to really memorize this slide. You especially don't need to memorize these concentrations until chapter 11, unless you want to, or you already do, because you've just seen this stuff before. So just recall that cells use these membranes to maintain gradients of ions and keep things apart. Okay. Passive transport. So here's a nice, maybe an illustration. If you had some sort of transmembrane protein, it's going to have on its exterior of this sort of cylinder, maybe imagine things that are hydrophobic to interact with the membrane, but it has some way to move charged molecules through, so maybe some charged stuff is facing the interior. Passive transport would allow the movement of molecules along a gradient from high to low. This stuff naturally wants to flow into or out of the cell, depending on which way it's going, to sort of equilibrate those concentration differences. To take something from a very low concentration and push it into a higher concentration, we're going to call this active transport. It requires some type of energetic input. And again, the details of this, we will spend more time talking when we get into later chapters. Okay. So if you want to move something that, say, has a positive charge, such as sodium or potassium, you would maybe need something that can selectively bind with these to get it to move across. Okay? So selective active transport is saying, oh, hey, I have a binding site that is specific to the molecule I'm moving. And so possibly something negative would be in here to interact with it. You don't need to memorize these examples, but we showed bacteriorhodopsin a few times as our sort of sample transmembrane protein. What bacteriorhodopsin does, its energy input is photon excitation or light. So that is the energy input it needs to move protons against their gradient. Okay? It's moving hydrogen. But you know, ATP is another possible source of energy input. You don't need to memorize these examples.